Hello, my name is Aaron Ellison, and I'm delighted to be speaking to you today on World Carnivorous Plant Day about some of the research that I've done with the pitcher plant Sarracenia purpurea, how we can use this research to better understand basic ecological principles and address pressing environmental problems, and how these really fascinating plants can be used to help others learn about ecology and the environment. I've been studying carnivorous plants since the mid-1990s, and all of this work has been done in collaboration with my good friend Nick Catelli, who is a professor of biology at the University of Vermont, and we work together with a large number of graduate and undergraduate students, postdocs, and some elementary school teachers too. You may be familiar with some of our work from two books that we've recently published. In 2018, I published the book Carnivorous Plants, Physiology, Ecology, and Evolution. This collection of technical papers written by over 60 scientists from all around the world is only the fourth major synthesis of knowledge about carnivorous plants and the first to focus on their ecology and evolution. You'll note that the second synthesis, by Francis Lloyd, was published nearly 70 years after Darwin published his insectivorous plants, which was the first one. The third synthesis appeared 45 years later, and ours only 29 years after the third. At this rate, the next one should be out about 10 years from now, and it is likely that some of you listening to this talk will write, edit, or contribute to it. Last summer, Nick and I published our Scaling in Ecology with a Model System. This is a much more personal book than Carnivorous Plants. While synthesizing our quarter century of research on Sarracenia purpurea, it also recounts how we came to study carnivorous plants and some of our adventures in the field. The research I'll tell you about today is part of two of these adventures. Our work with the northern or purple pitcher plant, Sarracenia purpurea, takes advantage of two important characteristics. First, unlike other species of Sarracenia, the hood of Sarracenia purpurea does not cover its mouth. As a result, when it rains, the pitchers fill with rainwater. In these little pools of rainwater lives a complete food web. This food web of bacteria, protozoa, rotifers, and larvae of flies depend for its energy and nutrients on the ants that are captured by the pitcher plant and that drown in the pools. The larvae of the pitcher plant midge tear apart these ant carcasses, and the pieces are then decomposed further by bacteria and pitcher plant mites. The bacteria and mites in turn are eaten by rotifers and larvae of the pitcher plant mosquitoes, which themselves are eaten by bigger larvae of a flesh fly. The waste products of this food web, the pee and poo of the bacteria, mites, rotifers, and larvae of the midges, mosquitoes, and flies, provides all the nutrients that the pitcher plant needs to grow and reproduce. In its structure and dynamics, the pitcher plant food web works exactly the same way as any other aquatic food web. There are bacteria, there are producers, there are herbivores and consumers, and there are predators. The other important characteristic is that there can be hundreds or even thousands of pitcher plants in a single bog. Each pitcher plant has a lot of leaves, and each leaf has its own pool of water and its own food web. So we can think of each leaf as an individual lake, and once we obtain the required field access and use permits from the relevant government agencies and landowners, we can do controlled replicated experiments on tens or hundreds of these miniature lakes at a time. This replication was especially important when we were trying to understand tipping points in lakes and identify early warning signals of these tipping points that environmental managers could use to keep lakes in good condition. In this slide, you can see two lakes, one of which is a healthy blue color and is very low in nutrients. The other one is an unhealthy green color. It is covered in algae because it has been polluted with excess nitrogen and phosphorus that comes from sewage, detergents, and agricultural runoff. Ecologists have been studying the processes by which lakes go from healthy to unhealthy states and looking for early warning indicators of this tipping point so that managers can intervene early enough to keep the lakes healthy. But these studies take a long time and it's hard to do experiments on large lakes. So we use the miniature lakes inside of pitcher plants to study this kind of tipping point. By feeding the plants different amounts of prey, we use dried and ground up bald-faced hornets, we were able to turn the clear pitcher fluid and its food web into a green, smelly, nutrient-rich cesspool. We were able to show that this really is a tipping point in the sense that very small changes in the amount of prey we fed the plants led to dramatic changes in the pools, and that these changes were difficult to reverse. It took only 3 days to tip the pitcher plant's pool into an unhealthy state, but even after 2 weeks, it had not yet recovered. Just like a lake, except that a lake might take years to tip from one state to another and decades to recover. At the same time, we used DNA probes to identify a set of bacteria that were good indicators of the healthy and unhealthy states of the mini lakes. We hope to use these DNA probes on real lakes and see if similar bacteria can be used as good early warning indicators of tipping points in lakes. We also use the miniature lakes within the pitcher plant to study how entire food webs change when top predators are removed. This is a major concern in the world's oceans, where we have been steadily fishing down the food chain, overfishing large fish like sharks and bluefin tuna that also are large predators, leaving behind only smaller fish and many fewer predators. Like tipping points in lakes, it's hard to do experiments in the ocean, but we were able to experimentally overfish the food webs in pitcher plants by removing the top predators. And then we discovered the real importance of these top predators in controlling every aspect of these aquatic microecosystems. The lessons we learned from pitcher plants can be scaled up to understand how fishing down the food web in the oceans affects more than just our dinner plates. It affects all aspects of the ocean's ecosystems. Now, we studied pitcher plants in New England, the northeast part of the United States, but these kinds of experiments can be done anywhere that pitcher plants grow, and even in your home terrarium or greenhouse. Because across North America, the same food web can be found in almost any individual Sarracenia purpurea plant. And similar food webs occur in the Asian pitcher plants in the genus Nepenthes. So this kind of research can be done anywhere else in the world too. And finally, this kind of research can be done by anyone at any age. For example, the Linnaean Society in London, through its Darwin-inspired learning project, has developed a series of modules for primary and secondary school students to learn about biology. Because Darwin wrote the first book on carnivorous plants, there are a series of classroom modules that use sundews and pitcher plants to learn about their ecology and evolution. So I'm glad I was able to share a little bit with you today about my research on carnivorous plants, and I hope that you're inspired now to go and do research on your own and learn more about these fascinating plants and maybe even change the world. Thank you very much for listening.
The International Carnivorous Plant Society wants you to be successful with your plants. We welcome growers just getting started all the way through professional scientists. We started an annual World Carnivorous Plant Day to celebrate these spectacular plants. Take a look around our website and you'll find historic documents about carnivorous plants, growing guides, free educational resources, and more. Have questions? Ask. We don't bite, but our plants do.